Good morning, everyone. I'm Derik Brown, the Senior Life Sciences and Diagnostics Tools Analyst at Bank of America. Welcome to our next conversation with Jason Kelly, CEO and co-founder of Ginkgo Bioworks, an innovative company here in Boston, who's going to talk to us about what Ginkgo's doing in their unique role in sort of approaching the cell and gene therapy ecosystem. Jason, with that, can you tell us a little bit about yourself? And for those in the audience that don't know what Ginkgo is, it's probably worth a little bit of a 101.
A little intro? Yeah, sure. Happy to do it. We're well lit up here, too. It's quite nice. Yeah, so good to meet you all. Jason Kelly, I'm one of the co-founders and the CEO at Ginkgo. A little background, we're located over in Seaport. Company today is, we're pushing about 1,000 people, about 250,000 square feet of really kind of robotically automated lab space, which I'll talk about, sort of one of the key assets of the company located here in Boston. We took the company public in October of last year, raised about $1.6 billion. We trade as DNA, the old Genentech ticker on the New York Stock Exchange. And the sort of core idea behind the company is to apply kind of manufacturing theory to the laboratory R&D activities associated with cell engineering. Okay, so we don't develop our own therapeutic drugs or end products. We don't actually manufacture drugs for other people. What we effectively do is have companies outsource cell engineering R&D to us, and we run it in a systematized fashion, which ultimately makes it much less expensive, so allowing for many more shots on goal, many more R&D attempts per dollar, and also in a way that aggregates data and learning across projects. And so this is a really important point, Derik, from my perspective. We do that cell engineering in really any market that's interested in genetic engineering of cells. So the origin of the company was actually not in therapeutics. So when we took the company public, right, we added, Ari Beldegrin to the board, from ex-Kite CEO, Reshma Kewalramani, current CEO of Vertex. So we are, over the last couple of years, moving more into therapeutics, and we'll talk about that today. But Ginkgo today has more than 100 of these R&D partnerships, $100 million joint venture with Bayer Crop Science to engineer microbes to produce fertilizer. We work in the animal-free meat space, making alternate proteins to go into sort of better tasting veggie burgers. We work in the cannabis space, industrial enzymes for things like detergents or fragrances and so on. And the commonality across all those projects is they need a lot of genetic designs to be produced, put into cells, and then tested to see which ones are the best for that application. And so that's really the heart of what Ginkgo is. And part of the reason we were so excited to build the company, my background, I did my PhD at MIT in bioengineering. One of the frustrating things I think about being a scientist, in bioengineering is you're learning all this detail about biology, how to design great experiments and ask key questions. And you've done that. You spend 5 to 10% of your time getting to think like that, and 90 to 95% of your time moving clear liquids around a lab bench by hand, right? And so you have this experience of like, oh, I have so much more I would like to ask, so many more scientific questions to ask, and the fundamental limitation is my two hands. And so we're breaking that paradigm. The scientists at Ginkgo is kind of the inverse. Yeah. They spend 90% of their time thinking about experimental design and the biology, and really we outsource that liquid handling into robotics infrastructure. So a little bit of background, but super excited to speak today about what we've been doing in the cell and gene therapy space.
Well, starting with that, what are, as your team looked at the cell and gene therapy process, what were some of the pain points? What were some of the tools that you thought you could address, you developed to sort of address some of these blockages?
Yeah, so we heard yesterday, I guess, from Mark Casper at Thermo, around some of the kind of general manufacturing challenges, and these CDMOs are leaning in to try to make it slightly better to do manufacturing in this space, but still very difficult. To me, this is unsurprising, right? It would be like if you were wound the clock to the beginnings of monoclonal antibodies or something, right? We should just expect that there is going to be both improvements on the physical hardware manufacturing side at CDMOs, but also accumulated learnings about how to actually design, again, the genetics of either the, whether it's the vectors or the capsid design or the producer cell lines. All of these things have upstream opportunities to be made better, hopefully in ways that end up being generically applicable across many end drug products, right? And so that's I think an important point I'd love to land with you all. We are making those sort of investments at Ginkgo into what we would consider to be general purpose technologies, right? So I'll give you some specific examples. So this is separate from, we have projects with customers. We work with Biogen on optimizing their particular AAV process. We've worked with Aldevron on, improving manufacturing of actinia capping enzyme going into nucleic acid products. We worked with a couple of the nucleic acid vaccine companies on optimizing their plasmid production process. Roche in small molecules, Selecta for, working on sort of next generation capsids for liver tropism targeting. And so we have like customer projects, but internally at the company, a couple things we're doing. We've been working on, in the area of CAR T, basically building out big libraries to explore, can we find new co-stimulatory domains in CAR T? And we're testing, to the tune, just so you get a sense of the numbers, actually millions of these designs in primary T cells. Okay, and so how are we able to do that? Well, we're actually leveraging infrastructure that we built for other projects, right? So we have, in-house at Ginkgo, the ability to generate these huge sort of genetic design libraries, and then we have all the automation to do the screening. Now, previously, to give you another example, for synthetic promoters. Okay, so we have a project, again, this is in CAR T, developing synthetic promoters where we're trying to improve specificity for different cell types, and indissolubility of these promoters. And so we did exactly that in the animal-free meat space, again, of all places, right? Where we had done huge promoter libraries to optimize promoters, in this case, for yeast producing like an egg protein or a milk protein, right? And so we had that ability to generate large libraries, put them into cells, run that test, and then we did just need to update kind of the downstream testing in this case to be sort of testing in these, again, in this case, primary T cells. And so that's an example, a couple examples, we can share some others as well as we go on, of what we would consider to be general purpose tools. So if you're a company that has a certain drug target and has a problem, that you think is a kind of a general issue, right? Whether that's the, size of cargo or things like that, we can work on the general problem associated with your specific optimization. And so we, with the capital we have in the bank, we can invest in that general purpose technology next to your specific drug asset. So that's a thing I'm pretty excited about as well.
So along those lines, so then what is sort of like the average, or do you have sort of like this metrics in terms of what the average cost saving is, time savings for when you do this? It's like, how do you sort of like measure, how do you measure success for this?
So it's really interesting. I mean, and this is obviously true in cell and gene therapy, but also kind of true across the board, I would say, in genetic engineering of cells. Overwhelmingly, what happens is these products fail to like get through the R&D process, right? In other words, like, we're not yet at the point where engineering cells is like the equivalent of building a bridge or a building or a software application, right? Where like the application and time and money with 100% surety will get you a bridge, right? The budget might go over, the timeline might be double, but you're gonna get a bridge. Not true, right? For many applications in genetic engineering, because there's sort of fundamental scientific risk. So actually a lot of what we end up getting metered on more is kind of, are the odds of success going up, right? Because when you actually get one of these products, it's a huge win, right? Because the industry is so bereft of actually having things come out the other end of the pipe, right? That's more the optimization, right? And so, but when we sit down, what we do is we sit down with a head of R&D, for example, at a company, and basically walk through the scale that their scientists would get access to by interacting with our infrastructure. And then they're just looking, okay, well, the budget's this, and their scientists are telling them, like, well, this would be 50 or 100 times as many genetic designs that we could try. Or this is a whole type of technology platform in the case, like for a lot of our, one of the ways we get to millions of samples is we do a lot of DNA barcoding and readouts on sequencers and all this fancy stuff. that might just not be available at all, right? Particularly, with smaller companies, right? But even with the larger companies, depending on where the technology's at. So a lot of times it's the researchers want access to some new tech, or it's a head of R&D saying, oh, this is a way to just give my scientists a lot more leverage than hiring a big, by-hand R&D team.
Yeah. So then how does the academic community sort of like leverage
Yeah,
this?
that's an interesting one. I'd be interested to talk to people about that. So far we've completely failed at doing that, is the short answer. We have gotten better at interacting with, we started off doing most of our deals with large companies. Again, not as much in therapeutics, that's been the last couple of years, but big ag, big food, big, like industrial enzymes and things like that. And so we kind of figured out over the last couple of years how to engage startup companies, right? So we do deals now that are, we're getting paid in equity. We're allowing them ways to like conserve capital because like, we want to give them the scale of this infrastructure. And like, importantly, Derik, and I'll get back to your academics question, but like, for a startup company, one of the advantages here is it's an ability to save on the capital cost of building out a lab. Right, so we have a company that launched on our platform. It's called Motif Foodworks. It's in the animal-free meat space again, example. They had raised, I think 50 or 60 million bucks. Launch, never built a biolab. Okay, so they built food science, like kind of applications testing, but they outsource all the biotech to Ginkgo. So they never had to hire, R&D scientists on the biotech side. They never had to buy a bunch of equipment and all the money it costs you to just have that lab in the first place, totally outsourced. Right, and the model that I would give you is, this is basically what like Amazon Web Services did for websites, right? Like you don't have to run your own servers. You should outsource that to big centralized facilities. Well, you've seen that in some aspects of pharma, right? You have the CRO industry. You don't want to run your own animal lab. You have the CDMO industry. I don't want to build my own giant tanks, right? Fine. Well, we're just saying, also when it comes to laboratory hardware, you shouldn't want to build that either. Right, like you should outsource that also to big centralized infrastructure, and that's a lot of what we're pushing. Startups benefit from that more than large companies, right? Because then they can save all that upfront laboratory build and scientific team hiring, and so I really like that. Now, if you jump to academics, I think right now it's still like the scale of a single academic lab is like their demand is a little too small to fit on our automation. That's the core issue. That said, if any of those areas, like I was mentioning, where it's like these general purpose reusable technology that would be useful across many therapeutic targets in the area, we'd be interested. If you're a lab working on that kind of thing, we'd love to work with you. We could sponsor research, we could get access, we could do things, co-develop things. We're really happy to give leverage there if it's a thing we want to work on, right? But helping you discover a drug in your lab, often that would be a smaller, your demand would not usually fit our capacity. But like, I'll give you another example, like iPSCs, right? So we've been doing, obviously challenges with something like a lenti or viral vectors as you're getting sort of random integration, right? So we have ways to target and deliver things, but where do you put them, in the iPSCs? So like doing a very systematic, hunt for good safe harbor sites where you have good expression, stability, safety profile. Well, we're doing these huge large-scale knock-in libraries now in iPSCs. That's another kind of general utility thing, and we probably could be doing that more with certain academic labs that want to ask those scientific questions, right? And I think right now that's probably just a miss on our part. So I know there's academic folks at the leading edge here in this room, so that would be something. Again, if these types of general use things are of interest to you, we would love to speak with you. Yeah.
So with just sort of thinking about the research outsourcing, I mean, it's been a long time since I've been in the lab, but I mean, scientists are very territorial.
Sure.
And certainly the ability to sort of control this, I mean, how willing are they to sort of like allow you to sort of like run with it and just do it? Just because you're wondering if they're sort of like missing, this is the competitive edge that we have from the science and like this and like this.
Yeah, so this is a super good question. So here's how I think it will play out over time. So that can be a challenge, right? Yeah. I won't dispute that. I think some of the things that we do see happen, like right now, for example, just to state the obvious, like there's like a capital crunch, in sort of like life science growth world, and so you see a lot of startups in the drug development space saying, hey, I've got a pipeline. Mm-hmm. I've got my lead candidate. I'm going to basically take the money I have and make sure I get that lead candidate through some milestones so that I can get to the next step on this thing. My candidates 2 and 3 are a little bit like, in a richer environment, I would have been pushing those hard. I'm going to keep them a little bit. I'm going to slow them down or put them on the shelf for a little bit. Well, those can be interesting candidates then to outsource. Okay, right? So we do see that sometimes. We see people go a little deeper, into the stack of things they're interested in if they are too sensitive. Because people also build relationship with us, right?
Yeah.
And see, and that's happened again and again. Like we work with a company no one in this room's heard of, a big fragrance company called Givaudan. And we started working with them on a pilot project 4 years ago. First very small thing. Now we've got 6 or 7 programs with them, right? And so that's the other thing. You build trust over time. That's one of the ways. But it's a real thing. Now the real hack is someday, I want you just to be able to log in to our platform and your scientists use it.
Right.
Right. So that it is kind of like just using Amazon Web Services. Like you've got your own people and they're just accessing the infrastructure, but you're really keeping the direction. Today it's a little bit early for that technologically. In other words, you actually have to be on site to really fully leverage our platform. So I can't offer that yet. But just so you know where I'm headed, that's where I'm headed, right? Like we get it. Like we're not, trust me, the majority of smart scientists do not work at Ginkgo, right? By definition. So we want to have all that leverage and push that back out. But today it is more of we work with you on what milestones you want to hit. We come with a joint scientific plan, and then our scientists execute it on our automation. But it's frequent updates and a lot of customer control.
So before I take questions from the audience on this, so then what are some of, if they're outsourcing to you, then what are the economics involved? I mean, is it a fee for service, or are you taking stuff on the back end?
Yes.
Yeah.
Great question. Yeah. So it's a mix. Yeah. So a typical deal, and by the way, there's knobs on these because different customers have different sensitivities. Okay, right? But a typical Ginkgo deal would include some amount of fees during the development process, right? Which again is usually, depending on what we're doing, somewhere between a 6-month and a 2-year process, right? So like take the project for Aldevron, right? They had this vaccinia capping enzyme. It's an ingredient into mRNA vaccines, nucleic acid vaccines. And so we improved over a period of about a year from start to finish that process tenfold. Okay, but through engineering of the microbe that produced that, and ultimately giving them a new manufacturing process. Okay, and so that was sort of a 1-year project. It included a project like that would often include something like fees during it, but then afterwards we get a royalty when they sell that product. Okay, and so but these are again, my mental model here is small piece of big pie, right? So because like I said, we want to be working basically with everyone. Right, so I don't need a big royalty. This is another thing that's a little bit different than you see in traditional therapeutic licensing, where I've got some asset, I developed it in my lab, it's precious, I've got 85 patents around it, and when I give it to you, you're my bet. And so I'm kind of very sensitive to the take. It's more like Apple App Store or AWS. I want to take a small piece, and ultimately, really, the advantage is we've now invested, probably about $500 million in just the physical infrastructure, right? And that's before you get to all the genetics. Like we make available all the genetics, right? So as all these projects we've done, we've been accumulating these reusable genetic assets, again, whether they're vectors, capsid designs, producer cells. You sign up as a Ginkgo customer, you also get access to all of that alongside the automation, right? So we're trying to do that, and then in exchange, take a small piece.
So any questions from the audience? So as we're sort of waiting for the questions to come in, I think a little bit more examples on what you've done with Moderna and Biogen and those other ones. I think a little bit more like that, just to sort of give the audience that might not appreciate it just a little bit more concrete.
Yeah, sure. So I can give you a couple examples on the therapeutic side. So with the nucleic acid kind of vaccine space, we've got a couple of these now. You mentioned Moderna's one that's publicly announced, but one of the areas here that, again, if you know how these nucleic acid drugs are made, you're basically doing a fermentation, typically of a bacterial cell, to produce a lot of plasmid, and then you're subsequently converting that DNA plasmid into RNA through a series of enzymatic steps, including some capping and so on as part of that. And so that first step of sort of optimizing plasmid production, we're very good at that. And so that's actually, again, it's not just limited to vaccines, right? It's really any step where you'd need to get a lot of nucleic acid out of that first bacterial production. We can do optimization both of the genetics, if that's something that's allowed. Okay, so this is where the regulatory rears its ugly head, right? So it's better to come earlier if you want to engage these next-gen tools, because once you get to a certain point, we're not able to actually, in some cases, people it's not worth it to change the genetics. And so we're just optimizing how you grow the cells, which we're okay at. But if we can change the genetics, that's what unlocks big things, more like the Aldebaran project. And then in the case of like Selecta, that's really exploring novel capsid space. Okay, and so they're really interested in like liver-tropic AAV capsids, and so we're looking to do basically, discover better ones there.
Got it. A couple questions for the audience. It's like, how do you handle intellectual property with your clients, partners?
Yeah, super good question, right? So top line issue, again, we have 100 programs like this, so we do this every day of the week, working with very big companies who are all sensitive to this topic, right? So here's the short answer to it. The product that the customer is developing, intellectual property wrapped around that is like the coin of the realm, right? Because that is what you need to defend copycats, right? And this is proven in pharma extraordinarily valuable. So all of that IP, so any IP for your end therapeutic is all exclusively licensed to you, rolled in, and serves as sort of a competitive moat around your product. The distinction at Ginkgo, as compared to like maybe a more traditional CRO, like you might imagine, is that we retain the rights to reuse the IP for other stuff. And this is why, we are able, like for example, as we've developed assets in the protein production space and industrials, well, now I'm doing tons of programs across the industry because we've been building up. We're just making it better. Like you work with Ginkgo now, and you actually have a shot at making a protein cheap enough to go into a hamburger, or into a animal-free yogurt or egg product. So like you have this issue in the industry where like you weren't accumulating enough assets to actually get products across the line. The only way we're able to do that is because we retain this reuse rights, again, for the non-product specific IP.
Right. So a couple of other ones. What is your repeat customer percentage? I mean,
That's a good question.
Are you doing one and dones, or people coming back?
It's a lot of repeat. But what I'd say is this: we have the general challenge with biotech, and this is like gets people confused also as they think of Ginkgo as a platform, is like the timelines, right? And if you look at like our rate of scaling, so in our automation, we've been roughly tripling the amount of work we've been doing every year for the last 6 years. And then alongside that, the cost on a unit operation basis in that like R&D factory, 50% cost drop per year. That means that in the last year, we've done more lab work than the first 10 years of Ginkgo, right? So we have this weird dynamic of like many customers who are in their like first 1 or 2 years. And then we have some of our older historical customers when we had less capacity who are the repeats. Okay, and but you don't really get the repeat business unless like people feel, doesn't mean the project has to work, by the way. People also get confused on this because it's still R&D. They have to feel like we were able to do something that they wouldn't have been able to do internally for that budget. And then we get repeat. But I mean, just given where we are as a company, it's more new business to be like in terms of like total number of new programs last year, more new. But if I look at our older customers, we're not doing bad on people re-signing up.
So what are some of the challenges and risks for continued growth of your business model? Question from the audience.
Yeah, sure. Good question. I think in new areas like cell and gene therapy, the hard part is just like proving the relevance of the platform, right? So like we're now proven on the industrial side. So like I have a lot of good demand there, right? And now it's more just like how can I make that more standard and how many programs can I add per year? In agriculture, we just announced we acquiring a big asset from Bayer, 175,000 square foot facility out in West Sacramento. So now we can go end to end to help ag biologic customers all the way from design the cell to formulate it to run a greenhouse trial. Okay. So in that industry, I also have like a lot of credibility now by virtue of kind of that asset and our previous work. In cell and gene therapy, we're proving ourselves, right? And so we have again we have work we're doing with Biogen and Selecta, a few other places, but like a lot of it is these programs we're doing internally. We're happy to share those results with you on sort of what we're seeing again with whether it's the safe harbor sites or the co-stimulatory domains or the synthetic promoters, and we're also happy to. Again, we just raised $1.6 billion. We're happy to invest our capital alongside your project to build out general purpose infrastructure of relevance. And so, but that's hard, right? Like when
Yeah.
you're not, when you haven't done it before in an area, people, you know, R&D is a critical industry, right? It's like, yeah, you know, prove it to me,
Yeah.
right? And so that ends up being one of the biggest limitations to growth.
I guess one other question that's come in is like, how applicable are your services for autologous gene and cell therapy products?
Yeah. So yes. So we would not be useful for like the manufacturing step on you know, in other words, like, oh, it's such a pain to like clone these things in. It is a pain, right? But that we would consider that manufacturing. Okay. In other words, like you know, the Thermos of the world, Danahers of the world, you know, Elevate, whatever. These people should figure that out. What I think we'll be good at is if you want to go after allogeneic, that is a very big engineering project. Like we're not there yet. And so that is an R&D where like I think the scale of the infrastructure we have could be relevant, and we're not actually yet doing anything big there. I think that's an opening if someone's interested.
Great, and we're right on time. And just want to say that next up, Dr. Christine Seidman and her panel will discuss the safety of gene and cell therapies. Thank you, Jason. Thank you, everybody, for listening to us, and enjoy the rest of the conference. Thank you.
Thanks, Derik.